This is Monkey Health Explorer. We're going to be talking about monkeys, although we won't be looking at monkeys, sadly. We'll be looking at their blood cells, which is really fun anyway, so works for me. So Monkey Health Explorer is a great project. We have done a couple of events with them, so you can find them on our YouTube channel with our SciStarter live series as well. They are big fans of 1 Million Acts of Science, and they've added already 3,478 acts as of yesterday. So we're looking forward to adding more to their list. They definitely need more impact here, and they have a specific protocol that they want us to help with. So we'll be doing a very specific part of their project. Now, to back up, if anyone is new to Zooniverse, this is a platform that citizen science projects use to develop a web page to essentially show a bunch of different images or scans or telescopic imaging and such to have folks like us, the citizen scientists, go in and classify images. So there's often way too much data that is too hard to get through, and so us as citizen scientists have to fill in the blanks and help them make the data a lot more manageable. So because it's Zooniverse, we have this lovely process to make sure it counts towards 1 million acts of science, and we have a leaderboard. So if you'd like to participate in the leaderboard on Zooniverse for today, it'll be live all of April. We have instructions for it here. So first off, if you're on SciStarter already, awesome. If you're not on SciStarter, the number one step is to make sure you have a SciStarter account so that we can count everything towards 1 million acts of science. You can also find a ton of other projects. Step number 2 is to register for a Zooniverse account as well. They're a partner organization, another platform, and it's really easy to register. I think they only use they only need your username, a password, and an email to make sure that you can get set up. And then once you've done that, you can add your Zooniverse username to your SciStarter profile, which is really easy to do. I'll actually demonstrate that for you. So my username, if I go to my profile here, my username is EC-Giles. So let me go back just so I don't lose that. So I'm going to go to SciStarter and I'm going to go to my dashboard. I'll demonstrate where that is. You'll go to your dashboard up here, my info and settings. I talk and work very fast, so I'm going to pause myself for a moment in case anyone is following along and is confused. I'm in my info and settings in my dashboard, and as I scroll down, I'll see this fancy thing that says affiliation integration. And so this section right here, affiliation integration, is where I can put my Zooniverse username, so EC-Giles is written there. Capitalization matters, case matters, so make sure you have the right username. And then I also have an iNaturalist account, so I have my iNaturalist username there as well. So if you want to do both, do both. If you have both already done already, awesome. That means it'll count towards this test tube automatically. So that's good news. You won't have to worry about reporting your acts of science at all. If you do have issues doing this, you can always report your acts here as well. Report acts, and it will take you to our reporting form for you to get moving on the citizen science acts you did. Sweet. All right, and with that, we're going to jump back over to, one more step. In order to join the leaderboard, if you would like to be on the leaderboard, you can also click on this link that I'll drop in the chat for you. It's also the QR code, which you can do Zooniverse via your mobile device too, so feel free to scan instead of do it on your computer. And this Zooniverse group is essentially our leaderboard, so if I were to click it, it'll take me to the leaderboard and we'll get to see who's participating over a range of time. So for example, on Thursday of last week, we had 432 classifications. Who was that? That's incredible. Oh, okay, well, that's amazing. So I think it was this person. Oh, wait. I think this person is here. Wow, incredible work. Awesome. Okay, so everything you do once you've joined that group means it'll log it into this leaderboard. So we'll get to see how many acts of science were done today, and also who's the winner of certain prizes, because like my t-shirt here, we have t-shirts to give away, we have hats to give away, we've got tote bags, we've got all kinds of stuff. So excited to bring that all to you. All right, so for the next 23 or so minutes, we're going to try out a project. Stop me at any point if you need me to slow down and help you work through it. I don't remember if I've done this protocol yet, so I clicked on single cell 2. You can also just click on classify and it'll send you somewhere. And so I've never done this one, so I have to go through and check the tutorial again. So Zooniverse goes through a tutorial, so you're trained on a very specific task before you start on the project, which makes the data quality better, and it makes you feel better about answering the questions correctly. So welcome to Monkey Health Explorer. In this project, we asked you to look at images of cells and identify the white blood cells by type by using example images in the field guide. So we will always have this field guide off to the right-hand side here where I can come back to it and check to see if I have the right answer and see if I can compare. You'll also see other things in the images, including many red blood cells. So we have neutrophils, lymphocytes, monocytes, eosinophils, basophils. That's the hardest part about this project. They're the B cells, the E cells, the M cells. You'll also see other things in the images, including red blood cells, round donut-shaped, platelets, small dark dots, oops, thank you, and bits of cellular debris. So there are a couple things, and I'm going to go ahead and take a look right now. So we got a neutrophil, which looks kind of funky looking. We've got a platelet that's like a little dot there. Lymphocyte has a big purple part to the cell, so it's almost like the nucleus takes up the entire thing. I'm making that up. I don't know if that's the nucleus. Red blood cells are the donut things. They do look like donuts. That's incredible. Okay. Eosinophils look similar to neutrophils, so that's a good thing to keep an eye on. It looks like they might be brighter in color, or it's noticeably only 3 circles inside. This one has a bunch of like bunched up pieces. And then a monocyte almost looks like a C. Hopefully that's the case for many of them. We'll see what the options are here. All right, so if we're new to Zooniverse, there's also some information here. I'm going to go ahead and skip through this one though, because I know how to use Zooniverse. So if we want to check out the different resources, we can come back to this later. All right, so how to identify the white blood cells. So we're going to be looking at the white blood cell in the center of the image. Choose the cell type from the list. So we'll have a list here to choose from, and we can use the field guide for examples to compare to. Pay attention to cell size. Lymphocytes are only a little larger than the red blood cells. So lymphocytes, let me back up 1, 2, lymphocytes. So lymphocytes are not that much larger than the red blood cells, which is good to know. Skip forward. Okay, great. Look at the cytoplasm. It is mostly smooth lymphocyte slash monocyte, or is there a texture? So is that a question? Neutrophil, eosinophil, and basophil are the ones with more texture to them. They've got more stuff going on. It's not just like one flat image or color, I should say. If there's more than one white blood cell, identify the one that's closest to the center. So we are only identifying one, which is nice. We can pinpoint. If there are no white blood cells, choose no white blood cells. Some cells won't exactly fit the description or match the guide images, so we'll just make our best guess, which is good. All right, feeling good so far? Cool. All right. And then there's some of these monkeys. So the monkeys we're looking at here are the type of monkey that are donating this blood for us. So in case you want to learn more about the project, I'll go ahead and go to about briefly just so you can see. This is about the rhesus macaque, which is an old world monkey. So we're looking at their blood cells because they're super closely related to humans. And so what we learn from their health in their blood cells will help us understand our health based on blood cells, which is pretty cool. Also, just look at how cute that monkey is. Just such a sweet monkey. It's a cute one. And where they live, they're out in Puerto Rico area, so you can read a little bit more about who they are, what they are, them hanging out on the beach, always, and then what this project is. I won't spend too much time on that because it's just a lot of reading, but if you want to know more, now you know where to find it. You can just go to About on the page on Zooniverse. All right, I'm going to go to Classify. If you're following along with me, I'm going to go ahead and send the link in the chat to join, and then on Facebook, I will do the same. Join the Zooniverse project here. Ta-da. All right, so let's get started. Okay, so on this one, I'm looking at this image. I'm going to go ahead and zoom in on my computer because it's a little hard to see, and I think you're pretty far away. So I've got this image, and the field guide is going to be helpful here, but also, also to note, we do have this lovely set of examples here, so we don't necessarily have to look at the field guide until we're not sure what to do. So for this one, it's very blurry, probably because it's very like I mean, it's centered, so they had to zoom in quite a bit. So I'm taking a look. It's got a lot of blue happening, but a lot of little pieces going on. It almost makes me think of a neutrophil just because it's got a little, like the texture is really chaotic. Chaotic. So I'm going to look at examples of the neutrophil. Yeah. I think it does look like a neutrophil because it does have quite a bit going on in there. So I might put that. Anyone disagree with me? Shout it out. Okay, cool. All right, we think of neutrophil. I feel good about that, so I'll go ahead and hit done. Ooh, okay. So this one, looking at the center white blood cell, I think automatically this looks like a lymphocyte. I would be surprised if anyone disagreed with me. It's an easy one, hopefully. And then I go for it and hit done. And then we're rocking and rolling, and then it goes a lot quicker. For this one, again, one that's kind of hard to see. It's got a lot of graininess on here. Maybe if I alter the image. That actually did not help, did it? Not really. For this one, it does still look kind of goofy. Got a lot, either a lot going on or nothing going on. It's just grainy. I'm tempted to put neutrophil, but I kind of want to learn about basophil briefly because there might be something to basophil being darker. Where did you go? There you are. Cytoplasm filled with large dark purple granules. Oh, that's what they're called, granules. Dark purple, bean-shaped. Doesn't look bean-shaped to me though. It just looks like it's almost looks empty. Okay. Yeah, I'm gonna go with it's not that. But the basophil is meant to be super large. Yeah, 2 to 3 times larger than the red blood cells. But I think it might be a neutrophil. So let's click on this and see 2 to 3 times larger than the red blood cells. Yeah, okay. So I feel a little bit better about neutrophil here. So I'm gonna click that, go back, and hide the field guide, and hit done. Going on to the next one. Identify the white blood cell in the center of the image, just like the previous ones. Okay, this one looks like it has 2 sections to it, so almost like a monocyte or a eosinophil. I'm going to look at the field guide for both of those. Monocyte, less common, so it's the least likely to be in here. It's bluish, very light colored. Irregularly shaped nucleus, sometimes C or bean shaped. That looks like it could be a bean, or at least it's a C shape, excuse me, not a bean. But it is a little bit challenging to see, so I'm not, I do think it could be a monocyte. Let's see what the other option is. Eosinophil, even less common. No granular, some vacuoles. Nucleus is dark purple, irregular shaped or bean shaped. So I think it could probably be either of those two. Eosinophil examples look like, looks very similar to this one. 2 to 3 times larger than the RBC. Was the size of the basophil? They're both larger. Oh, oops, wrong one. Wrong one. Monocyte, there we go. 2 to 3 times larger. So all of these are pretty large. Yeah, this one's tough. I think I might have to go to eosinophil. It is even less common though. Anyone have a particularly strong opinion about this one? No. Okay. All right, we're leaving it up to me. Oh no.
I feel like that one's, I'm seeing a C shape. I would go,
yeah.
I would go eosinophil.
So some C or bean shape, but I also saw that on the other one too, which is why I'm not sure.
Oh no.
Go monocyte. Yeah, sometimes C or bean shaped.
Wait, I missed that. I'm sorry. I
No,
saw
it's
C
okay.
shaped on the
I
other one. I was
wanted
like,
to
I
check.
got this.
Just to make sure you also saw this, but it does look closer to the eosinophil than it does to these examples. So I think I might have to agree with you. Maybe it's the gut feeling. That's what matters. Gut feelings matter. That is one of the tricks that in talking to Zooniverse project managers, oftentimes they're like, yeah, just use your gut, or like sometimes they don't know the answer, and so you're like, what? What do you mean you don't know the answer? And it's this is just a difficult task sometimes, and so it's not even that the scientists are better at it per se. They also are not sure sometimes. Okay, for this one, oh man. It looks like a lot going on, so I'm tempted to do neutrophil again. Let me double check that the neutrophil is, since the color is different, or the color is kind of different from these example images. Let's see. Most common, so not unlikely to have it there. All of them are C-shaped. They're so silly. Why even bother with any other descriptor? Okay. Yeah, all right, I'm gonna go with the neutrophil example. Neutrophil. All right. And then I go down and say done. Now, if I was super, super unsure, which to be fair, I probably could have done that on one of the previous ones. If I was super, super unsure, I can also hit done and talk and explain myself. Like, I'm really not sure. If someone has a different example or a different opinion, please write it in, because that is very helpful in cases of where there's an example image and there's conflict between the answers so that the scientist can look at it more specifically. If you're curious what's going on there, it's the dye that's used in the imaging. They had a spilling of the dye. For this one though, it looks like we might get lucky with it being a lymphocyte. You feel good about that? I feel good about it. Then I'm gonna hit done. This one, I see three granules of sorts. Was there one that had a 3-granule like eosinophil maybe? Oh yeah, I think that's an eosinophil. Yes. Sweet. Okay, and then I hit done. This is where I need like my background music, and then we just do crazy work while live. It's like a collective body doubling to be with each other while we work. This one is very funky, so I might actually think of this as kind of an error. So I'm gonna go ahead and mark it with what I think is best, which in this case, that might not even be a white blood cell, to be honest, because it's pretty small. So I'm gonna hit no white blood cell and hit done and talk, because this is a good example of this being a challenging one. Challenging image looks like there might be a mistake with the dye. Question. I classified it. That's not how you spell that. It as none. No white visible. All the sphere or all the parts of the image look about the same size. And then I add my comment. Boom. And then this will live in infamy until someone responds to me. Hopefully Marianne gets back to me. Marianne is the, she's right here. And Barrier Z, she's in charge of this program. Let me go back to Classify and see this one. All right. So for this one, similarly, it's got a bunched upness to it. So my thinking is potentially monocyte or eosinophil again. Let me use the field guide and remind myself about those two. So I've got eosinophil. It could be this. Could be. But could it also be a monocyte? So C-shaped and funky looking. Cell spread examples. Basophil. Very rare. Most common being neutrophil. Let me just double check that there's. Oh, you know, it could be a neutrophil. It's not very clear. Not very clear, but I think it might be because it's also the most common, so I wouldn't be surprised. All right, neutrophil, and then I go with next. Identify the white blood cell, of course, every single one. This one looks like a lymphocyte to me, 'cause it's got one giant granule. One giant granule. Ooh, a U shape. That's interesting. Might be more eosinophil. Let's see. Cytoplasm, light, typically bluish, no clear. Glandular, some vacuoles, dark purple, irregular shaped. Yeah, that looks like a eosinophil to me. Cool, moving on. And then it keeps going and going. And I might pause here for a moment. Does this, so I personally find this challenging because these are really close-up images, so the quality is a challenge a little bit. I know I've done some of their other protocols that might be easier to start with too, if you need practice, like this one, beginner's practice, where you are going through and actually just checking, practice identifying the white blood cells in the image shown here. It's just practice images, so we'll get to know if we get them correct. So mark all of them. If I marked this one, for example, I think this one might be a neutrophil. Okay, and then what about this one? This one also looks like a neutrophil. Then this one, which looks like also a neutrophil. And then let's see. This is a burst cell, which does not help us because we can't tell what it is. And if I hit next, let's see if we were right about those. I've checked my answers. Have I? Let's see. Eosinophil. Oh no, I got them right. Wrong. So neutrophil, we got that one right. Eosinophil though. So let me look at eosinophil again and see the comparison. Eosinophil. Light, typically bluish, clear, no granules, some vacuoles, dark purple nucleus, irregularly shaped. 2 to 3 times larger. Okay, so the big difference between eosinophil and neutrophil is what I'm struggling with. So let's go to neutrophil. Dark purple with multiple connected shapes. So maybe if it's the more complex it is, the more likely it is for a neutrophil. So the other ones where it's like clumpy but not entirely separated, that's where we need to be careful that they might be eosinophils, which, I mean, less common, but also there's 2 out of the 3 options in here, which is kind of crazy. All right, done. Let's look at this one. All right, so this one's a C shape of some sort, which is goofy looking. Looks like it could be eosinophil. Looks like it could be a monocyte. I'm thinking eosinophil. Over here, this is a very complex silliness. Too many little pieces. This one over here has got 2 sections, so I'll do eosinophil for that as well. Let me get out of there. Click. Oh, it doesn't let me. Okay. And then I think that was the only other one I had. Let's see. Eosinophil. Okay. And then, do we have any others? I think the rest are red blood cells. We got another burst cell. It's so pretty though. Awesome. All right, how'd we do? Eosinophil, neutrophil, eosinophil. All right, there we go. Now I'm getting them right again. All right. So definitely a good idea to do practice every time you do this, even though I've done this a couple times. So who knew? All right, I'm gonna pause here because we're about 4 minutes to the half-hour mark, and so my eyes need a little bit of a break. If you are doing Xenoverse projects, these ones can get, you can hyper focus on them, and so I end up doing a lot of staring without blinking. So that's my #1 requirement is I need to remind myself to blink every time I do Xenoverse projects.